So thank you for the kind introduction. And thank you also, Karen, for introducing, basically setting the stage on the whole epitope matching and molecular matching topic. So the title of my talk is Allele-Specific Solvent-Accessible Amino Acid Mismatches and their correlation with immunizer-specific HLA antibodies after kidney transplantation or pregnancy. So this is a collaborative effort, of course, and we're also quite lucky to collaborate with our Canadian partners in the NephroCage project on that part. But let's have a look what that's all about. So basically, we're dealing now with a very specific thing. So like Karen mentioned, like a gazillion of projects already that are all very specific, very precision and this is now one of these methodologies, let's say. So in terms of molecular matching in organ transplantation, we are at a point where we do know that molecular matching methods better predict donor-specific antibodies after transplantation than classic antigen matching or serology matching, if you want to consider that, which is still state-of-the-art, in fact. And we see that for various models. So we see that for the HLA matchmaker, which is basically the applet model. We see that for HLA-EMA, for the Cambridge model, and also for PERCH. So I just put some representative papers here, but there's tons of them showing that this is a beneficial approach. So taking a step back, why is that the case, or what are we doing? What are we looking at? I put this busy slide here in the middle, but we only need to concentrate on the center part, actually, which is the T-cell dependent B-cell activation. So we have the situation that we have donor APCs introduced by our organs, and they carry a whole bunch of different HLAs of the donor. And these intact HLAs are recognized by antibody epitopes on the surface. So the patient develops antibodies via this feedback loop here. And the interesting part is after receptor-mediated endocytosis of these proteins, these HLA proteins get chopped into small fragments. So by enzymatic processes, proteolysis, stuff like that. So all known onboard mechanisms of our cells, these small fragments then may get loaded into the HLA binding grooves, and from there on, the HLAs are loaded onto the cell surface where they are presented to T cells, and these T cells now may react towards these indirect T cell epitopes, which confirms the B cell that whatever it did in the beginning seems to be right, seems to be fishy, produce more antibodies of that kind. So you get a more and more specific antibody response over time. So it does make a lot of sense that this is in particular important for chronic allograft rejection, so long-term survival. So when talking about molecular matching, I mentioned there are various models. Question is, how are they different? How are they similar? Why do we have so many of them? Well, let's take a look into the details. So first up, we have the applet model, and they put here some models of crystal structures of an AO201 and highlighted an interesting position here on the 62, around 62, let's say, for the moment. So first, we have the applet model, which defines the surface of these HLA molecules into small patches with a certain radius, and if there's a polymorphic amino acid, considering all the different alleles, then you consider that as being a theoretical applet, and then you try to confirm that via monoclonal antibodies or certain, let's say, experiments to check whether that makes sense or not, and whether you can really dig down into the details to confirm that this must be the position, the polymorphic position here that causes trouble. As such, you get this funny nomenclature like 62GE, so two amino acids actually next to each other. For HLA-EMA, we consider polymorphic surface accessible residues, so they consider a neural network predictor, or two neural network predictors, in fact. predicting the surface of HLA loci and creating a map to distinguish what should be surface accessible. The EMS model is basically doing the same, but translating the amino acids' physiochemical properties into a map of charge, hydrophobicity, things like that, which is a little hard to to visualize. Therefore, I created that negative image here. But you get some sort of, let's say, map of properties of the HLA surface. And the perch model I just put here for to show that actually we are not only considering antibody epitopes when talking about molecular matching, but we can also consider the indirect pathway, which is covered by the perch model. So in fact, we are looking not for the intact HLA of the donor, but we're talking about processed protein, which is then basically a peptide being bound in the HLA binding groove. And forgive me, I put also an A0201 here just for the sake of them all aligning next to each other. In fact, you would see in practice a class 2 presentation towards CD4 positive T cells here. So these antibody epitope algorithms, and we dive specifically now into the epitope landscape, they assume that the structure of these HLAs is basically almost pretty much identical, which means the shape of the molecule looks the same. So HLA Matchmaker is doing that by manually annotating these structures, then assuming for other structures that the same amino acid configuration means there's the same exposed applet being shown. HLA Emma is doing that by creating these locus-specific maps of accessibility. So you consider that all HLA-As have the same surface characteristics, and basically, the EMS model also considers that to some extent by creating these, or by this translation mechanism of protein to charges, let's say. So you also rely on positions. The question is, how about treating these structures now independently? That's basically the hypothesis. And we tried that. We built this pipeline arbitrarily called Snowflake. Don't try to search any abbreviation for that. What we did here was to extract first and foremost the PDB crystal structures for HLA molecules. And then we decided to supplement these sequences by, let's say, meaningful and good predicted structures. And we used AlphaFold 2 for that, which is developed by DeepMind, which is related to Alphabet and Google. These predicted structures were then, got then a peptide docked into the binding groove, because naturally you will always have a peptide in the binding groove. So we needed to fetch known binders from the IDB, render the peptides in the binding groove with another predictor called ApeGen, and supplemented the data that we already got from the PDB. And this large data set we then analyzed for solvent accessible surface area by a rolling ball or rolling probe algorithm. So basically you roll a model over a three-dimensional structure, and where the model hits something seems to be surface. So this is our training data set for a neural network. You may say now, yeah, why you need that now if you have a pipeline to create a surface area for each and every protein. Problem is this right-hand side pathway here is quite time-consuming and computationally expensive. So you don't want to run that for each and every of these gazillions of HLA molecules that exist. So we implemented a neural network generator, fed the training data into it, and used it to predict the surface for each and every HLA, created an HLA surface database. So we are now able to provide an allele-specific solvent-accessible amino acid matching, so extending the previously mentioned methods. So just so you get an idea, we have these are basically the numbers of crystal structures from the PDB, and we are concentrating for this case now on class 1. Class 2 data will follow later this year. You see the vast majority is A0201s, so not very representative, and then we have a couple of others. And all the dashed bars here are basically the predicted structures. So you see we have quite a few A's, HLA-B's, and C's, and we only have few, actually just a couple of HLA-C's, which are coming from actual crystallography experiments in the PDB, so quite useful to supplement them. Problem is, of course, how do you compare these structures? Turns out this is completely non-trivial and completely counterintuitive, for me at least. So comparing the geometric position of the individual amino acids and atoms now just causes problems, because this is superimposition dependent. These molecules live in their own coordinate system, and as soon as you have some light difference in angles, you end up with a local optimization problem. So you overestimate local error in favor of a global minimization, and then the binding groove looks completely different in terms of mathematics, at least, or statistics, which in fact you would say is not the case. So an example here is in the purple circle, for example. These two positions here of the amino acids are pretty much at the same position. The difference here, however, is that there's a tiny angular difference in dihedral angles between the pink and the green loop here, and you see that in the purple circle, this does not make a difference at all. But the further away you get, the more and more this error adds up until the structures are different in terms of geometric position. But you would completely agree with me that the loop structure is completely identical. It's just shifted a little bit. Mathematically, that's kind of annoying to deal with. So we applied here two alternatives, which is compare the amino acids dihedral angles, and to compare the solvent accessibility as a functional difference metric. So what does that mean now? Well, we compared now all the A0201s pairwise with the other A0201s, all the A2402s with the other 2402s, and so on and so on. So comparing the structures pairwise within the same allele, and then we compared them pairwise, getting rid of all the locus information and allele information. So putting them all into a single bucket called HLA class 1, and comparing them pairwise. And if you check now the root mean square dihedral angle difference, so the how the angles change between the structures, this looks pretty much the same between, or if you check all class 1 pairwise compared to the A0201s, for example. So that means that there's basically no change or no difference between cross allele comparisons here. However, if you check now the surface area difference, there is indeed quite a difference between the pairwise solvent accessibility of an A0201 versus A0201 compared to putting them all into one bucket and, for example, comparing an A0201 with a B3501. So there is apparently some value in it. So that is now quite complex to understand, let's say, or to see what that actually means. So I pulled out one specific example here, which is on the left-hand side an A0201. On the right-hand side, we have an A0101 crystal structure, and I highlighted now the position 167. I don't know exactly which what amino acids that are, but it's position 167. And what you see on the left-hand side is that you would probably agree with me that this seems pretty solvent accessible or surface accessible. So an antibody should be able to dock that, right? But on the right-hand side, you probably also agree with me that this position 167 amino acid is very buried, and an antibody will have a hard time to dock to that. And that's exactly the point that we have proven now. So the accessibility of a position in one HLA does not necessarily mean the same position is accessible in another HLA. And in fact, our predictor here, called Snowflake, has shown that on the left-hand side we predict a surface accessibility, whereas on the right-hand side we don't. Therefore, we are happy to continue with that. So the next study we have carried out is a clinical validation in two cohorts. One is a pregnancy cohort that has been published well in the past. These pregnant women were NGS typed on 11 loci, and the child as well. And then we have a haplotype mismatch situation where the paternal haplotype is basically the immunizer here for the women, and they will form child-specific antibodies, and we screened for them in the post-delivery serum. Aside from that, I mean, of course, we are interested in kidney transplantation, but pregnancy is always a nice model system. But we also checked a kidney transplant cohort, which is a subset of the Berlin transplant cohort, selected for equal distribution across different allocation programs. So acceptable mismatch versus the regular allocation versus where no matching at all happens. So the old recipients, basically. All patients had no DSA prior to transplantation, very important. And we only had intermediate resolution HLA typing and imputed the rest by multiple imputation that also has been published on. And all of these patients were regularly screened for donor-specific antibodies post-transplant. So, long story short, we do see higher number of snowflake mismatches in HLA mismatched cases that developed child-specific antibodies / donor-specific antibodies. Let's take that synonymous for the moment. Makes life a little easier. So what does that mean? Well, actually, in all of these box plots you see, whenever we have child-specific or donor-specific antibodies formed, we see higher boxes. So all the right columns here are elevated compared to the cases where we did not develop a specific immunization. So we have HLA A, B, and C in the columns, and pregnancy and kidney in the rows. So that's quite promising. So we continued doing some regression analyses, and as we don't have a time dependency in the pregnancy cohort, it's just a let's say simple univariable regression model. We compared the different purge methods here just for the sake of completeness, or let's say because we could, and we compared that with antibody verified applets, as well as all applets, as well as the snowflake score. Now what you see is that indeed we have significant correlation between all of these metrics and child-specific antibodies, and on the right-hand side in a Cox regression model, the same for donor-specific antibodies for the kidney transplants. So this data here is already pooled by the different loci and scaled and normalized, so to compensate a little for the individual loci's characteristics, let's say, in terms of scoring distribution, but it's all still HLA class 1 here. So the initial hypothesis when we started, and that was basically the intention why I wanted to bring up the different modes of allorecognition again, is the initial idea was that the snowflake model should be a little more related to B-cell epitope matching than to indirect T-cell epitope matching. So what we would expect is that there is a lower correlation between the purge models and applets / snowflake, as well as snowflake and applets being a little more correlated. And therefore we calculated in this matrix here the various Spearman's row correlation scores, and you see if you compare the purge scores with one another, so the different presenters here being named, presenting loci being named, all class 2. Obviously, the correlation is not that strong, so between 0.36 to 0.49. If we check, however, the snowflake score now and compare that to the PERCH score, we still see a rather low correlation between 0.36 and 0.63 in the kidney cohort, which has the blue numbers, and the black numbers on the pregnancy cohort. And if we compare now snowflake with the applet models, we see indeed a higher correlation between 0.63 and 0.82. So in fact, that confirms at least a little our hypothesis that we try to basically predict the same thing, and seems to be somewhat the case, at least from a numerical standpoint. So, of course, the next question is now, how does that relate to the applets? Because the applet model is, in fact, established, of course. So we, however, also know that the model is not perfect. It's still a model, and there are still some open questions that we couldn't find an appropriate answer for yet. So we checked how does that relate to the applets now. And we have on the y-axis the solvent accessibility, on the x-axis the different applets, and we see here 3 interesting patterns. I don't want to dive into any applet in particular, but the patterns here are, I think, quite interesting. So, for example, we have this one here where we see, okay, a multi-amino acid applet. Some of these positions are not accessible, others are, but still quite conserved across the different positions. If we check now, or if we look for another interesting pattern, we see here that all alleles carrying this 143S applet are quite in line with each other, that this position is not solvent accessible, which is already inherently a funny thing if you consider that they should be accessible for antibodies. And then we have this third guy here that basically has a very widespread of the solvent accessible surface area of this amino acid here. So as an add-on, I created also the plot here for the non-antibody verified applets, because that is the obvious next question. We see here basically the same patterns. Again, I don't want to dive too much into individual applets here, but I think what's quite interesting compared to the antibody verified applets is that we find here quite a few of these applets that seem to be not accessible at all, which may be the explanation why they aren't antibody verifiable in the first place. But this is just an hypothesis, so don't nail me down on that. Right, so as you can see, it's like an additional metric for these applets, and I think from there on we now have a couple of ideas for experiments, what can be done next to complement that to the applets, for example. So speaking of next steps and concluding the whole presentation, basically. As said, I think there are now all kinds of fun experiments that can be done with monoclonal antibodies, or reanalyzing previous data on antibody verification to understand, to better understand these applets' accessibility patterns and why they are accessible, things like that. Of course, we want to extend that to HLA class 2, and spoiler alert, this will come this year for sure. Well, not this year. It's still 2022. Next year, 2023, it will come for sure. As well as a couple of improvements to the prediction pipeline that we already wrapped our minds around. Obviously, the ubiquitous question, how are these models all related with each other? Of course, this is one next step to link Snowflake and PERCH to aggregate both B-cell and T-cell molecular matching methodologies in order to, let's say, provide clinicians with one score and not like a gazillion of scores, and then we tell him, yeah, we don't know exactly how they are all related. They are somehow, but maybe just a little. And I think this is also a great thing that Genome Canada is doing. We need to find, of course, how can that all be translated into the clinics, right? So we need to find the answers, how that can work in an allocation algorithm, also with respect to fairness and access to transplantation. These kind of things need to be considered, as well as clinical guidance in immunosuppressive regimens. All kinds of funny little things that may follow now. So that's it. Thank you very much. Happy to take any questions from the audience. You're still on mute, by the way. Everyone happy?
Yes, can you hear us?
Yes, now I can hear you.
Oh, okay, great. Okay. We're just fielding questions right now. Sorry, I'll come over to this mic so you can hear me. We don't have any questions in the virtual chat. Do we have any questions in the audience? There is a question. If you could just say your name and where you're from, since we have such a diverse audience of listeners. Thank you.
Hi there, Ali Zaranpar, University of Florida. Since you spent all that time getting this thing to work, I will ask a question. What do you do in terms of when you model the proteins, right? I mean, they're not rigid structures. There's flexibility in there, and you know the solvent accessibility will change, right? So how do you propose to build that into your future models?
That is a very good point and falls under the improvements of prediction pipeline thing. So that's exactly what I always try to put everyone's attention to, because when dealing with people from the HLA field, they consider these structures quite rigid, but in fact they are quite wobbly, right? So we absolutely need to take care of these dynamics. And that's also what we learned already from the structured analysis of the PDB database, that we see quite some fluctuation in solvent accessibility across the different crystal structures. And that already indicates that these structures are not all completely 100% identical, and they are also flexible, which we also see in terms of B factor, for example, right? So very good comment, and that's certainly some next step.
Thank you so much.